afternoon to everyone. On the behalf of organizing committee, that is Indian Society of Weed Science and ICAR-DWR Jabalpur, I extend my sincere welcome to all the participants and distinguished experts and panelists on the 6th day of National Training on Smart Weed Management for Sustainable Agriculture. Today, our first expert is Dr. Gulshan Mahajan, sir. So before we proceed to our expert, I would like to give the brief introduction to our esteemed speaker, Dr. Gulshan Mahajan, sir. So Dr. Mahajan, sir, is a globally recognized weed scientist and agronomist with over 20 years of groundbreaking contribution to weed management and sustainable crop production. He worked as a professor at Punjab Agriculture University, India from 2000 to 2024. Currently, he is an adjunct faculty at University of Queensland, Australia. His research on weed control and water efficient rice cultivation has not only influenced national agriculture policies, but also benefited farmers worldwide. Dr. Mahajan sir is a fellow of National Academy of Agriculture Sciences, fellow of Indian Society of Weed Science, fellow of Indian Society of Agronomy. He also served as an associate editor of Weed Science Society of America. With more than 200 research publications, having an H index of 41 over 9,000 citations, his contribution have made a global impact in advanced sustainable crop production. Today, sir is going to deliver on the very important topic, that is weed biology and its importance in weed management. So the topic of immense relevance for modern agriculture and future food security. So sir, with this, I welcome you for this today's training program, sir. And now, sir, you can start your presentation, sir.
Thank you very much, but I am not seeing you guys here.
Yes, sir, JK Soni. Sir, here.
Can you hear us, sir?
No, I am not seeing you guys here. Maybe some problem, or I need to start again.
No, sir. I think your screen is visible. Your screen is visible. You can share your presentation, sir.
Okay. Then.
Sir, voice is coming.
Yeah. Everything is okay. So, is it okay now? My presentation is there or not?
Not, sir.
That's why there is a problem, I think. Okay. Now it's okay. So,
Yes.
okay.
No.
Not?
You can click
PPT, sir.
Yeah. We can see your screen.
Is it okay?
Yes, sir.
Yes.
Full screen, sir.
Full screen, you can go.
Okay.
Yes,
So
sir.
everything is okay now?
Yeah.
Okay,
Perfect.
sir. Yes.
So thank you very much, Dr. Choudhary, Dr. Soni. So I am very thankful to the weed science team of Jabalpur that they provided me opportunity for this talk on weed biology, the foundation for integrated management. So you talked about that regarding introduction. You talked about so much, but I am not like that deserved like that, because it's a continuous practice, and weed science research is a continuous, and nobody is a perfect expert in this field. So I am still learning a lot of things. When we talk about weed biology, so there are many students here. So let me first introduce what is weed. So everybody know that weeds are undesirable, unwanted plants growing out of their place and are hazardous to plant, human, and animal growth. So, in terms of layman language, that someone, for example, if you have a party and there is an unwanted guest in your party, and they eat like food, your drinks, and dance at the night. So, like that, the weed act like that, that they rob the nutrients, they rob waters, and then gave a competition to the crop. So what is biology? Biology is the definitely it's a life cycle of the wheat, that where how they germinate, how they grow, how much that at what time they flower, and how about their produce the seed reproduction system, and how they survive in the soil. So it's a very broad term, biology, and that's why that wheat biology, because if we understand the biology, we can identify the weak point in the life cycle of the weed, so that we can hit that weed cycle and make our weed management system sustainable. So that's why we need to understand the weed biology. Now the next question is, why we study weed biology? So you know that if you see on the x-axis there are years, and on y-axis there are number of unique cases of herbicide resistance. So you know there are more than 500 cases of herbicide resistance, and this problem is continuously increasing. And there are more than 270 weed species that has become resistant, and there are total 31 herbicide site of action, and among them 20 already tasked the resistance. And this resistance is a global phenomena, and you can see that it's spread over more than 75 countries. Now, if you see this slide, that on the x-axis there are years, and on the y-axis there are number of species. So if you see the red, this one, so red, and this one blue. So this is group 2 and group 5. So number of resistance cases in these type of herbicides are more in group 2, and second is followed by 5. So group 2, which herbicide are? Sulfonylurea, like IMI herbicides, so they are group 2. Now, why this problem is more in group 2? Because they have more high selection pressure. You know that case of isoprotein resistance against Floris minor in India. So this herbicide that it was like it worked for a long time in India, more than 15-20 years when it was introduced. But when the new herbicides like Clodinafop, leader or sulfonylurea type of herbicide, when they were introduced in late 90, so after 4 or 5 years they become resistant. So the question is that if the herbicide is having a high selection pressure, more effective, more will be the persistence, and early will be the resistance. So this need to understand. We need to understand this phenomena of the herbicide. Now, so if you see that resistant species, so now there are many terms you already that in the previous lecture, I think there was already you learned about the what is cross resistance, what is a negative cross resistance, what is multiple resistance. So if you see that in case of Lolium rigidum, that is annual ryegrass. So this weed is having like Poa annua also. This is having 12 number of resistant to 12 number of site of actions. It means that it has a multiple resistance. So see that if there is a multiple type of resistance, there are some weeds like Lolium, Poa. So we are very concerned. Now, Lolium rigidum annual ryegrass is a number 1 problem in Australia. Very difficult to control. If you see that recently we uh, uh, did some studies that this herbicide is resistant to the glyphosate, haloxifop, and clethodim, and many number of herbicides. Because the stacked resistance genes are there. Now, why the question is need to understand that why this problem is more in case of lolium. So some weeds having a specific their growth characters. For example, lolium is having a very high affinity for cross pollination. So with the weeds that have a very cross pollination type of behavior, uh, weeds that have a more fitness penalty, so they become more problematic in the for resistance. So very careful if the weed is having a multiple herbicide resistance. These are very difficult to control with the chemical type of practices. See the scenario of glyphosate against annual ryegrass. So if you see that in 2021, so resistance percentage was 8%, and within 3 years, that 21 to 24, the resistance cases against annual ryegrass increased to 39%. So that's why that if you herbicide is having that some weeds are having multiple herbicide resistance, so it's very difficult to control and the resistance problem continuously increasing. Now, second question is why to study weed biology. Weeds, you know, already caused economic losses. There are many economic losses. And if you see this scenario, this is the latest figure that weeds that caused almost 34% yield losses. And if you compare with insect pest, it caused 38% of yield losses. And on the right side, if you see annual revenue losses from weeds in terms of Australian $1 billion per year. So there is a annual loss of, if you compare Australia with India, so 3 times more losses. So this is the, I think, data collected by Jabalpur, and there is around 15 billion Australian dollar per year loss due to weeds. And if you compare with Australia, it's around 5 billion per year. So, and so you can see that magnitude that how much that weed causing losses to our system. And in terms of grain production, Australian grain growers, so weeds are causing 4.3 billion losses. Now, see the scenario in terms of cost per hectare. So this is the figure from the GRDC. GRDC is our Australian, this is like Mandi board like in India. So this is a grower board, and grower board means they fund they funds for research there. So they estimated that there is a total cost that due to weeds that increased $206 per hectare, and that cost includes that yield losses $27 per hectare and $176 per management cost. So the message is that weeds cause economic losses due to the yield loss or due to the increased that production cost. Now, not only that economic losses, like weeds also caused environmental losses. So if you see the red circle here, so these are the red circles. You can see throughout the world that due to the pesticides, these are the figure that there are more that environmental losses in these areas. Particularly, for example, if you see the scenario in India, so northern side is because they are taking more herbicide use, and Punjab, Haryana, and also cotton crop. So there is a more that environmental losses there. So why these losses? Because the half the spray is wasted. And if you that see the scenario in Australia, there is about 13,000 kilometers of river that have unsafe level of farm chemicals. So this is the recent report in India. I could not find this type of data. And overall, if you see that globally, 2 out of 3 farm are at risk from pesticide pollution. So this is the latest report of the Nature, published in Nature. Not only this, in climate change also, because weeds are very prone adapt climate change and become more competitive. This is I am sharing that it's just a 2-page that summary in the current science that published that how we respond to climate change. So if you see that if you increased carbon dioxide and under the global warming status, that most of the weeds that C4 weeds are more beneficial, but it is not always true. C3 weeds can also benefit. Like if you see that weedy rice, this is a C3, so it's benefit more than the cultivated rice because it has a more plasticity. So similarly, in case of like if you have a drought condition and like in winter, Phalaris and Vina ludoviciana, they have a competitive edge with the wheat. So if there is a erratic rainfall, drought or rainfall, there will be multiple weed emergence and reduce the herbicide effectiveness due to the seasonal change or something like that, because you need to control for long term and herbicide will not work. And that's why we are very much concerned in the wake of climate change. That's why we need to develop integrated weed management strategies to handle, and we need to understand the biology of the weed, particularly in the wake of climate change, so that we can develop sustainable weed control uh, for that scenario. This is a simple example of Australia. You know that buffalo grass, and the buffalo grass, it thrives under hotter, dry, and fire-prone area. And fire is a very frequent phenomenon in Australia. So it's in a vision driven by the climate-related disturbance. Similarly, rigid ryegrass and hairy flea bean, so they adapt quickly to warmer and dry environment and posing increased management challenge. In India, you can take the case of Parthenium hysterophorus. So under elevated, you can see that it was earlier that Parthenium was problem in the plain, and now it's problem increasing in the mountain area like in Himachal Pradesh or Uttarakhand. So you can see that due to the climate change, that the weeds that have a more seasonal plasticity, they can adapt to another environment, another regions. And now again, take the case of like management practices also. For example, if you are shifting from transplanted to direct seeded rice, so the weeds that were problematic in the maize like love grass, or you can find that Isamium rugosum. So these type of weeds that are in the dry area, so they can become prominent due to the environmental change, due to the agriculture change in agriculture practices. So shift phenomena, the weeds very adapt quickly, and because of plasticity. So we need to understand, that's why need to understand the weed biology. Now this is the slide that a few weeds that we definitely need to understand, because the weed biology, because these are the main worst offender for best result. For example, if you see that in wheat or in India, wheat and rice are predominant crop. So what are the weeds in the main weeds in the wheat crop? Like Phalaris, in rice, like Echinoclone, Rascally. So at least you must have the knowledge of main weeds if you want that are predominant in your main crop. And for example, Phalaris, Echinocolo, Amaranthus in cotton in India, Chinopodium. So at least you must have the knowledge of the biology of these weeds. And like in Australia, these are the annual ryegrass, wild radish, and feather road grass, wild oats. These are the predominant weeds in Australia. So at least we must have some that whole knowledge of these weed, so that we can develop sustainable weed management strategies for these weeds. So again, I am concluding here that why weed biology matters in IWM. So there is again a small paper. If you want, you can read this paper. So message is that herbicide resistance cases are increasing, particularly if the weed has a fitness penalty. So we are more concerned about its biology, economic losses, environmental degradation, and climate change. So these are all points that we need to, that how weed responds to these changes, and that's why we need to study weed biology. So if we if we have a knowledge of these type of knowledge on weed biology of these weeds, we can enable targeted, timely, and sustainable weed control. We can reduce the cost, delay the resistance, and improve our weed control. Now the first step in weed biology, that we must have the knowledge of the how to identify the weeds. We must have a knowledge that how to identify that type of particular weeds. And it's very difficult. One can learn by experience, particularly when the weeds are very small. So very difficult, but there are some cues you need to understand. You need to have knowledge of that cues. Now here I am citing two examples. For example, wild oat in wheat. It's very difficult to that identify that when these are at the seedling stage. Okay, but there is some cues, like if you see the seedling leaves of the wild oat, they twisted anti-clockwise, and the wheat, the leaves are twisted clockwise. So this is the point that to identify that type of weeds. Now again, that Avena fatua and Avena ludoviciana, these are two species and very difficult. You can't identify at the seedling stage for these species, but there is a cue for its identification at the maturity stage, and which is Avena ludoviciana and which is Avena fatua. So Avena ludoviciana seeds, they always shatter in pairs on the ground when at maturity, while Avena fatua, they shattered singly. If you see that these are two awnts, these are two seeds that are united together. I am not seeing here. Can show. Oh, sorry. What happened?
Sir, click center and move, sir. No problem.
Not.
You want to play video, sir?
Yeah. Okay.
So
So if you see that Avena ludoviciana, so these are two seeds that are united together. So in Avena fatua, there is one part that shatter on the ground and one part remain on the plant. So this is the point that you need to catch that point. With the help of that, you can identify which is Avena fatua and which is Avena ludoviciana. Now the second example, that cultivated rice and weedy rice. So there is again that you can differentiate that the cultivated rice, it always short and uniform, And weedy rice, if you find in the field, it's very tall and variable. And seed have white husk. And if you compare with the weedy rice, it has always on and rats and shattering tendencies. They are very prone to shattering. And leaves and panicle, light and compact in case of cultivated rice, dark and open in case of weedy rice. And if you see maturity, it's very irregular in weedy rice, and it has a very strong dormancy. But again, some of the that races of the weedy rice are different. It's very difficult to identify, but again, with experience, you can learn more things. Similarly, for Phalaris minor, how you can identify at seedling stage in wheat. So if you see that pinkish liquid, if you squeeze that the base of the plant at seedling stage, the pinkish liquid squeeze from the base of the plant. So this is the point that you can identify Phalaris minor in wheat. So you have to understand that how to identify the weeds at early stage, because otherwise, if you are not identifying the weed, so it's like that a heat a shot that in the door. So whatever you are doing, for example, if you are using herbicide, if it is not recommended for that weed, so there is a wastage of herbicide. So that's you must have the knowledge that how to identify the weeds in the crop or weeds. Now again, the weed life cycle. So you need to understand that weeds are annual or biennial or perennial. So annual means they complete the life cycle in one season, biennial two seasons, and perennial means that for the more than 2 years. And perennial weeds are more difficult to control because they reproduce vegetatively and by seeds. For example, take the case of sorghum halicans. Now we have a GRDC project in Australia. So what we found that the farmers were complaining that sorghum halicans is having a resistance against dual gold. But when we did study and we did not find that any resistance in case of sorghum halicans. But then again, we analyzed critically that why the farmers are reporting, because uh, it's reproduced by vegetatively and by seed. We were working with the seeds, not by vegetatively, and it has a rhizomes, and if they grow by through the rhizome, so it's very difficult to control. So that's why the farmers are reporting, because in their field the sorghum plants are that growing through the rhizomes. So that's why these are very difficult to control sometimes. So you must have a knowledge of weed biological traits also. For example, because now the new practices are coming. For example, laser, thermal, electrical weeding. But for that application of that technology, you must have the knowledge of the weeds. For example, uh, which weeds having a meristem position open. For example, broadleaf having a meristem position, they on the upper side are they are exposed meristem. So in that cases, you can use the laser and thermal technology very well. So and if you take the case of life cycle, as I earlier told, that annual weeds are easy to control than the perennial weeds. Now take the case of electrical weeding. If the weeds have a more moisture content, so it's very easy to control with the electrical weeding. And for example, why? Because the conduction is more in case of that, if the weed is having a more moisture content. And again, take the case of parasitic weed. You must have the knowledge that which type of biochemicals that these type of weeds having. For example, in case of striga, there are different peroxidase or sometimes endoglucanase and cellulase enzyme. They break the cell wall in the parasite, and Historia enter into that one. And now see that CRISPR technology is coming. CRISPR means they are silencing the gene, host parasite relationship. So if you have the knowledge of the weeds, uh, you can guide, you can give your feedback to the molecular biologist that okay, these type of things are in these type of weeds, and if you can silence, you can provide the feedback. If you can silence this type of gene in the host plant, so these weeds that we can develop a resistance crop against these weeds. So already work is going by ICAR, I think, for Striga, and it's in the progress. Now, critical period for weed control. So this is also very important because it's a balance point between the weed biology and crop biology. So you must have to know that in which crop, what is the critical period of weed competition. For example, in wheat, critical period of weed competition is different, 20 to 45 days. In sugarcane, it's a 30 to 120 days. So you need to define the critical period of weed competition, because if you apply herbicide after the critical period of weed competition, you can't get the good weed control. So critical period, the message is that critical period of weed competition, these influence the herbicide efficacy, and for that knowledge of the weeds is very much necessary, because critical period of weed competition also differ with the dormancy pattern of the weed, with the emergence pattern of the weed. So that's why critical period of weed competition is very much that you need to study when you are working on different type of weeds. Now again, the seed germination ecology. So it provides that if you know the germination cues, you can get the basic knowledge that how to control these type of weeds. For example, see the case of this. These are 5 on the x-axis. There are alternating 5 temperature. Why? This is a Australian case study on barnyard. Why we selected 5 temperature here? Because Australian, one thing you must understand, Australian climate is different than of India. When in India there is a winter, in Australia there is a summer, and when there is a summer, here is winter. And especially in Queensland region, the climate is very much similar to the northern India. So they have 4 seasons like summer, winter, autumn, and spring season. So why we selected here, we according to the that type of temperature, that 35 daytime temperature to 25 night temperature, 30, because it's a summer weed. So we selected 5 temperature from winter to summer. So what we see here, that we have seen that we know that it is a summer weed, it responds well in the summer temperature. So you can see that higher germination in the when the temperature is higher. So but if you see light, dark and dark, so when we tested in dark, the germination is reduced under the dark. Okay. So the thing is that if you get this cue, you can apply this cue to in the practical sense, because you know the idea that under dark environment germination is reduced. So you can increase the you must have that if we bury the seed in the soil at deeper layer, it will not germinate. If you have a residue cover with mulch, it will germination up to some extent can be reduced. See in this case that seed burial depth. So when the depth is increasing, the germination is decreasing. So we must have the knowledge of these type of studies on particular weeds. Then we can apply this knowledge into the practical sense. Here it is osmotic potential. We know that it love moisture, this weed, Echinochlora colonna. So we tested at different osmotic potential, and we have observed that 50% germination reduced at minus 0.44 osmotic potential. So these type of knowledge that we must have. For example, if we see that okay, at high osmotic potential, this weed is also having a higher germination. It means this weed can germinate this in the drought environment and give a strong competition to the crop. So that type of knowledge that we can gain from these type of studies. But again, that I think it's unfortunate that we have this is a very basic study, but we need incubator, and if we have incubators, and if we study these type of weeds in the incubators should we work at the it should be system should be on the generator. If there is abrupt of electricity, you can't get the real results. And automatic day night temperature, it must have a timer. So these type of facility you have, if you have, you can do these type of studies in your lab. Now again, the similar case that in African turnip weed. So this is a winter weed, and if you see that again under the dark environment, the germination reduced. It's a winter weed, so you can see that germination reduced significantly with the dark environment. And when we tested again under the residue cover, if we increase the residue cover, the germination reduced. So these type of cue we can get from doing these type of studies on the seed germination ecology. But again, these things did not work for every weed. Now take the case of this one. This is our latest publication coming on wire wheat. So again, in this weed, under dark environment, germination is reduced. If you see that under emergence, this concept hold good here also. If you under dark germination, percentage is decreasing. Under burial depth, percentage is also decreasing. But if you see in this one that under the residue, if you increase the residue 0, 1, 2, 4, you can see under the if you don't have a residue, germination is just 20%, but under 4 residue ton, the residue germination increased. Here also in this population, these are the population. So in this population, it increased. But what we understand that germination reduced under dark, germination reduced under burial depth. But why this concept is not applicable here? Because some weeds behave differently. If you have a partial residue 2 or 4 ton per hectare, the concept that some light reaches that they activate the light diffused and they activate the phytochrome, and as a result, and when the other conditions are favorable, temperature is favorable, moisture is favorable, so these have a more germination there than because the residue also increase the conserve the moisture content. So favorable environment is there, so that's why here the germination is more. That's why each particular weed is important. The concept you develop on one weed, it may be applicable or not be applicable to the other weeds. So we must have knowledge of each weed specifically. So message is that we need to understand the biology, whatever the weed that we want to control specifically. So weeds are successful competitor due to their biological traits. For example, if the having a small seeds, it's easy to control because the small seed only on the surface, mostly on the surface. And if you eliminate that either with stale seed bed, either with chemical control, and don't allow the seed production, you can control easily. But in case of large seed, they have more longer persistence, particularly if they are buried, and burial depth increase the persistence. If weed emerge at the same time with the crop, they are more competitive than the others. And if the emergence shift is more, very difficult to control. And seed dormancy diversity, because if there is a dormancy in diversity, then there is a staggered emergence. So for staggered emergence, you have a that long season weed control, and it's very difficult. High seed output when you have a high reproduction potential of the weeds, so definitely some weed will survive, and they will go into the seed bank, and you that long term weed control is difficult. Wide dispersal mode, if they have a wide dispersal, they can spread from one place to another place easy. Adaptation to climate, because weed have a more plasticity, so very much that very quicker in adaptation. And herbicide resistance evolution, uh, particularly it's very fast in the weeds with a shorter life cycle. And if it has a less fitness penalty, low fitness penalty, then again it would be a severe problem in that field. So seed dormancy mechanism, so it's very important. If the seed has a very high dormancy, it means it has a more persistence in the seed bank, soil seed bank. And there are three type of dormancy, like physical dormancy, physiological dormancy, and combinational dormancy, means having a both physical as well as physiological. So physical dormancy we can now, why the dormancy? Why we need, if the weed have a dormancy, it's very difficult to study the biology itself also, because for biology study, if you can't have a 90% germination, you can't do these type of experiment, light, dark, these type of osmotic potential, residue cover, because for these germination ecology, seed must have a more than 80 to 90% germination. So if the seed is having a very high dormancy, it's very difficult to uh, get the knowledge, biology knowledge on that weed. So that is the also point. So physical dormancy, as you can see in case of hypomia, so very hot seed coat, and it can't take the water. And physiological dormancy, sometime inhibitors are present in the wheat seed. For example, every seed has a GA and ABA balance, and if the ABA is more and GA is less, the seed that even if they are in the favorable environment, they will not germinate. And see the case of annual ryegrass, that they have inhibitor, flourish minors, they have that phenolic acid, and once it in the rice wheat system, then the phenolic compounds they are released, and then when the winter comes, and they germinate. So some have a combination dormancy like Indian hemp, and they have both physical and physiological mechanism. Hard seed coat and inhibitors are present, and when they release, and then they germinate. So more the dormancy, more the persistence. So, but this persistence also increase with the boreal depth. Now, this is another study that we did in the Australia. So we buried the nylon bag at different depth in the soil, and then exhumed that nylon bags at different intervals. So this trial we conducted at 3 locations. One location was at Gatton and Narrabri, that is in the New South Wales, and St. George. So, and this Gatton is that having a high rainfall area. St. George is a drought environment. But if you see that 90% of seed decay, so here you can see that in the surface they decayed in the 7 months. But here at St. George, they decayed in the 11 months. In the drought environment, they decay is fast where the rainfall was more, and decay is less where the rainfall was less. So if you see that at shallow depth, the persistence was that 15 of 90% of seed decay after 15 of months. But in case of St. George, where the drought environment, that 90% of seeds decayed after 17 months. Now the question is why here the acts on the surface Gatton why the decay was fast on the surface, and at sand zones, why the decay was slow at sand zones. So what it indicates? Indicates that soil and climatic conditions, they affect the persistence status of the seeds in the soil. For example, Gatton has a more rainfall and less clay content. Here we have that sand silt clay content. If you want more detail, you can read this paper. And because there was more aeration, so wheat germinates and decay was fast. But here it has a more clay content, and seed will there was anaerobic condition, seed remain in the soil, and that's why the persistence was more. So message is that wheat can behave differently at different places. So soil and climatic condition, not only genetical, soil and climatic condition also influence the persistence of the wheat seed. So we need to do these type of studies in different type of environment, in your own environment. The result maybe it's not that result you may get at Jabalpur. It may be applicable to the northern India. It may or may not be applicable to the northern India. So these are messages that you have to develop the system. You need to study under your own condition that what is the persistence of this weed. Now see again the germination and emergence pattern of wild oat. Now see this one, we buried the seed. We use when the season, for example, wild oat. So when the wild oat mature, when the seed shattered on the ground, at that time we took that seed and we buried on the surface. Okay, and then we evaluated the emergence pattern at different time interval, that when they will germinate in the next season. So here the environment is like that May, June in Australia, this is a winter season. Here May, June are the summer season. So don't confuse with that one. Okay, this is a winter season wheat. And what we observed from here, that if we bury the seed, they started to merge in March 2000. This experiment started in November 2017, and after 5 months they started to emerge when the autumn rainfall was there. So we observed that wild oat emergence varied with boreal depth and seasonal condition, and emergence from 5 centimeter. If you see this right green lines, so here about this is the March month. So we started to emerge from the March and from 5 centimeter depth. And if you see that 0 and 10 centimeter, the emergence peak was lower. So it means less emergence was there. Okay. And peak emergence occurred at 5 centimeter. This is 5 centimeter, and peak emergence, if you see that in case of May, June and October, it was more. So the message is that this weed is showing seasonal expansion and demands extended period of weed control. Means this weed, so these are the message that weed started to emerge in March. For example, if you sow the crop in April, peak is here in the May or June. So we can adjust our planting that either after this period, or we need to that if we sow that in the May or June, we must have a strong weed control. And if the weed again emerging in the October, so we must have like a sequential type of herbicide treatment, so that can provide long term of weed control. So from this type of knowledge, so you can make your strategies for the sustainable weed control. So that's why the weed biology knowledge is must. Now again, the seed bank ecology. Again, you know that the seed bank ecology knowledge is very much required. Once the seed, if you see that this is the soil seed bank, and seed plants mature, shatter seeds, and it goes in the soil seed bank. Some are decayed with the help of microbial decay. Some are germinate and fetal germination that are buried in the soil. Some decayed with the abrasion of the weather, like sometime light, hot and cold environment, and some germinate and decayed. And there is a predation. Some are sometimes there are ants, so there are rats, so they eat some of the seeds. So ultimately, that you have to reduce that this seed bank, and for this you need to reduce the seed rain. Means you need to restrict the fresh seed production in the field, and for this you must promote harvest weed seed control technology. If the seed bank is there, you can make strategies for summer fallow, or with the help of stale seed bed you can control that large seed weed seed bank in the soil before the emergence of the crop, before sowing crop. And this is the ultimate example that of annual ryegrass, which have a multiple herbicide resistance, that how this annual ryegrass within 3 years reduced without that by adopting these type of techniques. What they did, they used a rotational beginning with autumn hay. They sow the oats, and before setting seeds, they plowed that they mow the crop, and at that time, annual ryegrass also mowed at that time, and then they grow the field pea with post-emergence lithodim, and because they used they collected the peas, and they used the post-emergence lithodim that control the early flushes of the annual ryegrass. And after taking the peas from the field, again they did the crop topping with the help of paraquat, non-selective herbicide. And you can see that this is a what was the annual ryegrass. There are more than around 5,000 seed production was there per meter square, and it reduced to the 200. So with these type of technology that you can, that if the if the weeds are having like a resistance problem, so by making that type of strategy program, you can control the weeds within 3 years. Now prevention is better than cure, and you know that in Australia you can't enter into the grower field without permission of the growers. And in every paddock, in every field, there is a message, come clean and go clean. And there is a heavy penalty if you are spreading the seeds from your vehicle to their farm, and if they have seen, and you may get a high penalty for that one, either economically also legally also. So there is a set policy, but in India no such seed policy is there. That's why we are struggling here. So the question is, that message is that there should be that we should seed should not be returned from the one place to another, and prevention is the best method. That if you restrict the entry from one field to other field, you can develop a better weed management strategies. This is the harvest weed seed control practices that are very prevalent in Australia. And these are the innovative techniques that are developed in Australia. And this is the only technology that spread from Australia to USA. And now USA is also that doing these type of things, and preventing their seed production with the help of chaff lining. So there are 3 that harvest weed seed control that developed by Australia. Chaff lining. So they make a channel chaff, including wheat seed into a narrow line behind the harvester, and for decomposition. And when there is a chaff line, they can control. When they germinate, they can kill with the herbicide, non-selective herbicide, or sometime with the decomposition, they also that the seed get desiccated. And this is the seed destructor. These are the seed mill, like a Harrington seed destructor. They crush the wheat seed. When they harvest the crop, they also crush the wheat seed in the with that combine. If they have a Harrington seed destructant attached to the combine. This is a narrow wind warning. They concentrate residue and seed into burn strips, and this effectively destroying up to 90% of the viable seed in the dry condition. So these are the technologies that are very well they are using. But again, this type of technology you can't do for all type of weeds. So that's again the knowledge of weed biology is must. For example, when you come harvest the crop, if the weed seed already shattered the seed, there is no mean to use these type of techniques. Okay, so you must have the knowledge of seed retention capacity of the weeds at the time of the maturity of the crop. So that's why, for example, now see this is again our study. See, this is Avena fatua and Avena ludoviciana. Okay, so if we analyze the wheat seed retention capacity of the wheat at the time of when we harvested the wheat. Okay, so in this wheat there is a like 70 to 38% wheat seed retention capacity, means Avena fatua seeds already shattered. Most of the seed already shattered when we harvest the wheat crop. But here in Avena ludoviciana, see that this is 80%. If you have infestation level of 3, 6, so there is 80, 75%. It means if you have this type of intensity in the field and 80 or 75% seed still there, seed retention is there, so you can use that type of technology like harvest weed seed control in your paddock. So this knowledge you must have that what is the retention capacity of the weed when you harvest the wheat crop. So again, it means you must have the knowledge of weed biology. Now again, high fecund and high reproduction potential. So almost every weed, if the weed having a high reproduction potential, shattered on the ground, okay, shattering capacity is more, like in a Vena fatua, this like in jungle rice, again Echinoclea colonna. So this is, you can see that if they merge at the 0 week after emergence, 2 week after emergence of the crop. So if they merge with the crop, the seed production is potential is high. In case of like a Echinoclea colonna in sorghum, and if you see the case of feather finger grouse, and you can see that again the seed production potential is high. So if weed merge with the crop or within very short time with the emergence of crop, they have a high seed production potential. So this knowledge you must have during that time, that for example, that's why biology knowledge you must require, because if they have a early weed emergence, it must have a high seed production potential also. So if you have, for example, if weed are emerging late, so does not have that high seed production, so you can escape the herbicide. So that's why the weed knowledge is very much required here. Now see the weed dispersal mechanism. So weeds are very good traveler. Okay, so some are like you know that some are like a free entry with the seed, crop seed. Some go with the some sit on your tractors, your implements. So there are 4 ways. Like some are blown by winds, like a feather top, road grass, sonkas, flax leaf flea bean. So they are blown by wind. See the example of flax leaf flea bean. It can blow more than 10 kilometers. Okay, so it can spread if they have a like a feather or something like that, ants, they can wings, so they can move from one place to another place. Now water, they can, if there is a flood, now you can see that in the northern India is highly affected by floods. So water is coming from the Himalayan to the plainland, Punjab, Haryana. So already many of seeds, they now flow from that area to this region. So that's why you must have knowledge of for example, if Jabalpur if DWR can plan like survey what was the status of particular weeds in that area at this time, and what could be the status after the this flood. So you must gain that type of information. How many new weeds introduced from the Himalaya to the plain region? So these type of studies you must be done here also, if the particularly for the that weeds that are blown by water. So machinery is a very that you when you used implement, when you reached machinery from one farm to another farm, again there is a movement of the weeds from one field to another field. Take the case of annual ryegrass, particularly if the weeds is having a multiple herbicide resistance. So this is a very serious concern. Animals, some you know that from animals in Australia, it's a very common feature that they graze the land and animals are in the field. They move from one field to another field, so they get in the reproductive system also, and from there they can enter from one field to another. So some like a feather top road grass, so they by if there are sheep, so they can move by the via wool. So there are different type of studies that have been done in Australia. So the answer is that we must have the knowledge of weed dispersal mechanism, that how much weeds moved from if there is a fire, how much moved from one area to another, if there is a flood, how much they move from one area to another, if there is a machinery movement from Punjab to Rajasthan, how many seeds are moving from one area to another area. And this is more problematic when the weeds are having a herbicide resistance. This is the case study of machinery that you know that this is the Australian example, that in Western Australia, grain guard, there is a protocol. Protocol means they must have, every machinery should come in the field as clean, then contained, and they confirm the, for example, they have 4 component here in Western Australia grain guard: clean in and clean out. Bring the machinery clean, and when you use for the another time, then clean out. Okay, clean before entry. There must be dedicated clean down, regular wash down, target seed trap, record and verify if there are seeds in the machine. You need to record and verify, and there should be a accountability. Quarantine suspect load, avoid cross contamination from one field to another field, prevent recontamination when you are moving from one field, and you must have the verify that it is not contaminated with one seed from one field, not contaminated with the weed seed. So without grain guard here, if you see the relative wheat spread, there is 100% seed spread, and with grain guard and 80% reduction was there. So this is another study of the Michael Walsh that was published in 2010, that how machinery help in dispersal of the seed from one field to another field. Now once you develop gather the knowledge of weed biology and how you can implement that knowledge into the integrated weed management practices. So now the slides. Now I will explain with the suitable examples. Now the question is that all the integrated weed management practices, these are not the true integrated management practices. For example, if you mix the if you may in your treatment make like a herbicide plus 1 hand weeding, 2 hand weeding, these are not integrated. Integrated weed management that are permanent, that you can apply in the field. Balance between weed biology and crop biology. So here the crop competition plays a major role in developing an integrated weed management practice. Now I am citing examples. See the case of this one. This study was published in Agronomy Journal, and so here the T-way interaction is there. Day, sowing date, normal seed rate, higher seed rate, and these are the 4 weed control practices, herbicides, and these are study was conducted that for the integrated control of wild oat in wheat. So if you see this normal planting, 7th of May in Australia. So if you have an effective herbicide, low seed rate, okay, so there is a gain of yield because you control the weeds very effectively. But if you see the case of low weed control, like in non-treated control, with increased seed rate, yield is increasing. Here, the poor weed control was there in case of Treflan and prosulfocarb plus acemetolachlor. This is a ready mix herbicide. Weed control was poor, but again, when the weed control was poor, there is a increase in yield with the increasing seed rate. Okay, but in delayed planting, if you see that either you used low seed rate or higher seed rate, the yield was similar in all the cases. So there is no advantage in this one. So that's why that if, for example, this now see the case of better you can understand when I will show you the V data. So the question is need to understand if there is a like with shifting of planting. For example, if wild oat in India, if you see in northern India, wild oat relatively prefer warmer temperature, while flourish they prefer very cold temperature. Okay, so if the infestation is more like wild oat, you can delay the planting. If the like infestation of flourish minor is more in the field, so you can do your planting little bit early, so that at that time when the weeds germinate, the crop ultimately that suppress, they will develop the canopy cover and closer canopy earlier and then suppress the weed. So you can design your technology on the basis of that what time they emerge. So you can develop that type of develop technology on the basis on the by knowing the knowledge on the biology of the weeds when they prefer better emergence time. Now see in this paper that when you see the weed data, if you see that here is a cumulated effect that averaged over herbicide. So here is the sowing date and seeding rate interaction. So you see that if you increase the seed rate, so there is a no effect between the. So if you see that there are two messages from this slide. One is that with higher seed rate, you can decrease the seed production. Either it is early normal planting, either it's a late planting. And when you increase the seed rate, the seed production significantly decreased with the lower seed, if compare with the low seed rate, but there is a no effect of the date. So that type of knowledge that you can gather from this type of information and then control the weed effectively. Now in this case, sowing date and herbicide interaction. If you see that here weed control was poor with the because in control you did not use any herbicide. Here the Treflon, the herbicide did not affect the weed. Poor performance here was poor. But here you can see that with delayed planting, you can significantly decrease the seed production. Here also. But when the herbicide were effective, the difference between the dates was very much less. They are statistical at par. But if you compare with the poor weed control and early date planting, so weed seed production here reduced significantly. So this is the integrated weed management practices, like you can control the wild oat effectively. So the message is, if you use more like not rely on the herbicide, use many hammers, there will be less selection pressure and the problem of herbicide resistance can be delayed. Now see the again that this is a study on direct seeded rice. It was conducted, the experiment was conducted at Ludhiana. So here you can see that when you use the higher seed rate in direct seeded rice, the wheat density reduced significantly in all cases. Gross wheat density in each variety, the wheat density reduced significantly. For example, if you compare this one, and again similar trend. Okay, but here there are three scenarios. Here it is variety. Here is a weed scenario, weed free and partial VD. Partial VD where we used only pre-emergence herbicide. And here you can see the effect of variety. This is DAN 202. You can see that under weed-free condition, either you increase the seed rate, either decrease the seed rate, the yield is same. And even under partial VD condition, the yield is remains same. Means this variety is a competitive. Okay, but in this case, like it's a common thing that if you have a weeds in the field, yield may be reduced. So similar trend is here. Under partial VD condition, every variety is losing yield. Okay, but here in real 367, there is a no difference between increasing seed rate, but under partial VD condition, there is an increase in yield. So means this variety real 367, so it is more competitive, and you can these type of material you can include in your crossing program for developing competitive variety. Again, that knowledge, that stage of the plant, like if the herbicide is effective. For example, here again the same message that higher seed rate decreasing the Avena ludoviciana infestation. Okay, weed seed production, weed biomass, and weed seed production. So another thing that we applied Noxadan at Z12 and Z33 stage. So these are the phenological stage. These Z12 means 3-leaf stage. Z33 means little bit after maximum tailoring, almost 7-8 leaves are there. So you can see that if a herbicide is effective, you can delay the application without compromising yield. But here you can see that if you apply the herbicide at Z12 stage or Z13 stage, so at this stage, still that you can reduce the seed production negligible. For example, if you apply, you can even control the late flush. Okay, if you delayed the application of herbicide, provided the yield is not compromised. So knowledge of the weed, that which weed you can apply that particular herbicide at a later stage, and even then it killed the plant. So it means you can delay your application. So again, that there is example of no-till and conventional tillage. No-till, some surface seeds, if the seeds are on the surface, particular are wild oat, then you can adopt the no-tillage practices and control the wild control effectively. And this technology also hold good for the flourish minor also. For example, if you do no-till, for example, now the resistance problem that came in it was very alarming during 90. But we introduced the zero tillage practices, and ultimately the resistance problems that we, with the help of this technology, we got a very good results at PAU. This is a excellent example
Sir.
of.
Sir.
Yes.
Actually, we are running late, sir. So can you?
Okay, 2-3 slides. 2-3
yes,
slides.
Yes.
So this is again the annual ryegrass and chickpea. This is a classical example. For example, we have a 2-row spacing here, 2 varieties and 3 weed situation. That wheat introduced at 0 stage, early stage, 3 weeks after emergence of the crop, and 6 weeks after emergence of the crop. So this is a simple that when you use the broader, wider spacing, 75 centimeter, less competitive variety, and you can weed intensity was more. And with a single change, either you change single variety, single row spacing, or changed kept the weed free for first 3 weeks, you can significantly reduce the seed production. When you used two changes that combined with the change variety and row spacing, the again change variety, early weed control, and when you adopt in the 7th scenario, changed variety, row spacing, and early weed control, so significantly reduce the seed production. So this is the message for the integrated weed management, that how to use that one. Again, this is a mulch, that this is a recent, that how you can use the mulch and significantly control the weeds, particularly grass and broadleaf weeds in moving crop. Now the herbicide resistance rises in India. In Australia also we have a heat tolerant sorghum. So question is that when you introduce that type of new practices, you must have to use the conventional herbicide in that one. For example, if the herbicide resistance problem again came against that type of particular herbicide, if you have a, you can use into your herbicide strategy program and reduce the selection pressure. For example, we are using atrazine in case of hemi-tolerant sorghum also. So rotate the herbicide to reduce the selection pressure. So that's a stewardship guidelines are must when we are using like any hemi-tolerant or something like that. We need to rotate the herbicide, and that's why integrated knowledge of weed biology that design strategies that maintain herbicide effectiveness for longer period of time. So weed biology. So in this, the ultimate conclusion is that know your enemy, strike when they are weak. Okay, by knowing the weak point in the life cycle of the weeds, and mix it up, rotate tools and tactics so that weed can't outsmart you. Stop the seed production, think about long-term control strategy, protect your crop, keep your crop more competitive, adapt to change if the weeds are shifting with the climate change. So you need to change your practices, protect your land, and keep the soil, water, and biodiversity healthy. And now the question is why these weed biology not common, that weed biological issue. What are the issue? Because some farmer are using that, it's some researcher, while funding is very poor in these type of issue, because some are seeing that it is too academic, focus on quick fixes. Some want herbicide give quick results, and weed biological this is just confusing, and this is long-term practices and complexity of information. And when the ultimately herbicide, people think that when the herbicide available, why do you use this type of technology? Sometimes economics also work in that system. So that are the issue that are related to the weed biology. So this is all about, and thank you very much. I very well, and I acknowledge that especially Dr. Bhagrat Suhan, that who provided me opportunity to work there, casual staff, GRDC, who provided us funding for doing these type of works, and PUU Ludhiana, that provided me opportunity to work in Australia, and opportunity to work at PAU, so that I can with the help of that I was able to develop this type of technology. And thank you very much. And now I'm very happy to answer your queries and questions if there is any.
Thank you so much, sir. One person, Anita Mahapatra, has raised their hand. So, sir, we will take one or two few questions, sir. Anita, can you ask?
Yes.
Anita Mahapatra. Okay, I think she is not there. So in chat box, any question is there? Just a general question, sir, by Sakshi. So if we will use all the principles and methods of weed management for control of 1 hectare weeds to reduce 100% to 50%, how much cost will require?
How much? In weed biological, you don't need any cost. These are the just practices that are already you are doing. So only need to implement. For example, if you are going for the narrow row planting, So you need, but things are that you must have that type of system there. For example, if you want to that reduce from 50 to 25 centimeters, you must have the drill or you must have the system. Okay, seed rate like you can use, there is a little bit cost. Okay, spacing, time of sowing, there is no cost in that one. So you can implement, you can, but I never that recommending that you can escape from the herbicide. You need to integrate with these type of practices, and ultimate aim is to that make the crop more competitive against the weeds.
And one more from Madhurima Pauri. Can you unmute, Yananji? Madhurima?
Yes, sir. Hello.
Yes.
Sir,
Yes,
am I
yes.
audible? Good afternoon,
Yes,
sir.
yes.
Thank you so much, sir, for explaining the topic so clearly. So, I wanted to ask a general question as a young researcher. So, if we wanna go through with biological researches, what will be your suggestion to the young researchers so that we can more focus on what are the important areas that we should be focusing on as a young researcher? Please suggest some.
Yeah. So first you need to understand that the proper facilities is there on if you want to work on weed biology. For example, we must need incubator. For example, I just giving you a example that when I was previously last year I was in at PAU. I took a retirement last year. So I brought the incubators. You must have 4 or 5 incubators there, and this should be connected with the electricity. Okay, generator, so that they should not be abrupt electricity if you want a focused and very good result. And thing is that you can each that incubator must be set at a different temperature. So if you have a proper facilities there, proper for example at PAU I brought, but their timer is not working. Day night temperature automatically should work. That 12-hour light period, 12-hour dark period, and temperature may be changed alternative. If you have facility, then you can select the weed biology work. If you don't have that type of facilities, then it's very difficult, and sometime that the results whatever you will report, so there may be some issues and during the publication. So see your institute, but this is a very good that if you can, even sometime I recommended that why are you for DWR, why are you investing more in these physiological, and if you are doing work on physiological type, then provide the incubators to the institutes so that they can develop that type of knowledge. I think Dr. Mishra is already working on these lines, so definitely they will grant in the CRIP and provide the facilities to the different institutes.
Okay, so with that, I think no more questions are there, sir. So with that, I am very much from on the behalf of society and from the Directorate of Weed Science, sir, and from myself, I am thankful for sir for nice presentation. Definitely, weed biology, understanding the weed biology is very much important for effective weed management, sir. So, sir, with that, we are moving to next presenter, sir.
Thank you very much. And if you have any questions that you can later through email, you can communicate to me, and I would be happy to answer these type of questions.
Surely, sir, we will come back to you, sir.
Yeah. Thank you. Thank you very much.
Thank you.
Okay, sir. Thank you.
Bye.